Hello, hearing scientists. I'm Dr. Susan Nitrower, and this is a lecture on speech perception and hearing loss for SPA 3032, Fundamentals of Hearing. So with that said, let me go ahead and share my screen here. Okay, I'm going to share the screen over here and begin the PowerPoint. Okay, so by this point in the course, we have covered the auditory system, and we now realize that the auditory system does much more processing than just transmitting the signal from the periphery to the central auditory system. We also have talked about the speech signal, the acoustic structure, and we realize that linguistic units, especially phonemes, are not represented in the acoustic signal of speech as discrete, neatly organized units. Rather, some sophisticated perceptual processing is required in order for the listener to recognize that speech signal. And that's especially true in difficult listening environments. Okay, so now the question is, what happens in the face of hearing loss? And here we will define hearing loss as raised auditory thresholds. So let's move on here then, and I'm going to give myself this laser pointer. Okay, so before we get into talking about hearing loss and speech perception, let's just go over the audiogram, which is used to benchmark or to metric the degree of hearing loss. Also, since we have been so interested in cats in this course, I wanted to give you the opportunity, in case you worry that your cat just never listens to you, you may give your cat a hearing test. And here is the website for giving that hearing test. Be forewarned that the frequencies that are presented in this hearing test are ones that humans cannot hear. So be careful about that. Make sure that the sound on whatever transducer you're using is turned down initially. Okay, so moving along here. Here we have frequency on the x-axis and sound pressure level on the y-axis, a spectrum, if you will. And we have the minimal auditory field and minimal auditory pressure functions here. So again, minimal audible pressure is what we get when we test thresholds, auditory thresholds, with a headphone or an insert earphone. And so they're monaural. And you can see that this is what we get with the minimal audible field. You can see that we have slightly better thresholds here, right? The thresholds are slightly lower. And why is that? Again, minimal audible field measurements are made with the speaker. And so both ears are operating and we get slightly better thresholds. And you'll recall the term binaural summation. Whenever you have two ears, you get better thresholds than when you're measuring just one. And either one of these functions shows us that our hearing is most sensitive in the mid-frequency range, in the range of thresholds associated most strongly with speech recognition. Okay, so now here, this is how a typical human being can recognize speech, and it's shown in sound pressure level units. But as we've discussed, it would be very difficult to be consistently describing hearing loss using these units, because you always have to be thinking, oh wait, is 50 dB at 100 hertz, what does that mean? How serious is that? Or versus 50 dB at 1,000 hertz, okay? So some nice scientists about 100 years ago gave us the audiogram, in which now we've flipped the values. Now we have 0 at the top, so hearing loss becomes poorer as we go down the graph. And now 0 dB, this is hearing level, not sound pressure level, hearing level. And 0 dB represents the quietest sound that a typical human can recognize at each of these frequencies. Okay, so again, in terms of sound pressure level, 0 dB hearing level is a different value at each frequency. Okay, so this is the basic audiogram. You in your work might once in a while come across the banana audiogram. I am not a fan of the banana audiogram. Banana audiogram shows this section in the middle that is shaped like a banana, and on it, some well-meaning clinician or someone put where they thought you needed hearing in order to recognize each of these phonemes. Being students now of speech and hearing science, you now realize that recognizing speech sounds or phonemes is more complicated than this, and that there are many cues spread across both time and the spectrum that allows you to recognize any one phoneme. It also has on here conveniently where other sounds are located. And you can tell this is a slightly older banana audiogram because nobody has phones like this anymore. And in fact, even lawnmowers, now most of them are electric, so not so loud. Okay, so that's the banana audiogram. You might see something like this. And now you can begin to see that in terms of hearing loss, what is considered a normal loss, a mild loss, a moderate loss, and so forth is beginning to show up. You can also see on here that hearing loss for children, it goes from being, the range of normal is less than what's considered normal for adults. It only goes to 15 dB hearing level, whereas for adults it goes to 25 dB hearing level here. This represents the fact that even a little bit of hearing loss for a child can have profound effects on their ability to recognize and develop speech. Okay, here's another example of what you might see. Again, here we see the normal range. Sometimes you will hear losses between 15 and 20 or 25 dB referred to as minimal losses, okay? And then mild hearing loss goes to about 40 dB. Then we get into moderate loss. Usually moderate loss is considered 40 to 70 dB. This audiogram puts in here moderately severe, and then severe is 70 to 90, and anything over 90 dB loss is considered profound. So about here, it used to be that losses greater than 90 dB were indicators that the patient needed a cochlear implant. That criterion is creeping up. Certainly now, any loss greater than 70 dB could spark the recommendation of a cochlear implant. So when I said you need a cochlear implant if you have losses greater than 90 dB for sure. Okay, what are you going to actually see if you become an audiologist or a speech language pathologist or an attorney who works with individuals who have hearing loss? You're going to be looking at audiograms that look like this. Okay, and I guess this goes also if you're going to be an occupational therapist or a teacher or anyone else who might act with individuals with hearing loss. So this is what you'll see. This is a mild hearing loss. The red line is the right ear, the blue line is the left. Red round right is the rule. Okay, so the red round line and symbols always represents the right ear. So here you can see this is a mild loss, about 20 to 30 dB, a little bit higher in these higher frequencies, a moderate loss around 50, 60 dB over here. And you can see the speech bananas lurking in the background. Okay, here we have a severe loss. Okay, so you can see here, this loss thresholds are well below the speech banana. So what you will hear is that means we need to amplify this individual's hearing so that these lines, when amplified, can get well above the speech banana. Here we have a profound loss. The right function here, the function for the right ear, indicates what we call a corner audiogram. This is very common. Sometimes patients it's almost considered a tactile response here if you get slightly better responses in these very low frequencies. Okay, and then finally, this is a pretty common audiogram, especially for older patients. As we're going to see, age-related hearing loss takes hearing away in the high frequencies before the low frequencies. It's also indicative of an individual who might have a hearing loss due to chronic otitis media as a child, or ototoxic drugs. They all affect hearing in the high frequencies before the low frequencies. You will recall that high frequency sounds are coded at the basal end of the basilar membrane. So any infection that can get into the cochlea across the round window there can affect those hair cells at that front end or the basal end of the cochlea. Okay, you're also going to see audiograms like this, where the C represents thresholds, aided thresholds with a cochlear implant. Now here, this doesn't tell me whether this is 1 or 2 cochlear implants. This shows me thresholds for unaided left ear here. Okay, here's another example of what you'll see. So here, aided thresholds with a cochlear implant or 2 cochlear implants, it doesn't really tell me. They all sit comfortably above the speech banana. So that means this individual is getting sufficient amplification. Okay. And then this 35 dB sound reception threshold, that just means that at 35 dB, this is a child. This child can hear speech at 35 dB hearing level. That does not mean the child can understand that speech. Okay, let me just check here. I want to make sure that, okay. I don't know if any of you ever had the opportunity to see the Flintstones. There was a cartoon that was around when I was a child, and it was based on an older show called The Honeymooners. But anyway, Fred Flintstone was actually a bit of a Neanderthal, which added to the humor. But this little YouTube video illustrates for us what various degrees of hearing loss does to the speech signal and to our ability to recognize it. So here we go.
1, 2, 3, 4, 5, 6, 7.
Aha! You're on my apartment building on Granite Avenue. You owe me $300. Get it up?
Fred, take it easy. It's only a game.
Wilma, I'm just like them big tycoons. I play to win. Now, Barney, pay up or get out of the game.
Put the bucket.
That's 1 down and 2 to go. Betty, it's your turn.
I don't have any more money either. You got it all.
Then I'll take a mortgage on your open home. Well, come on, take the dice, will you? Don't just sit there like a dummy.
I will not have you talking that way to our guests. Come on, Barney. I think we'd better go home.
Okay, So that gives you a taste of what the various levels of hearing loss does to speech recognition. Now let's talk a bit about the various etiologies of hearing loss, the various causes of hearing loss. First, let's talk about genetics in hearing loss. A genetic condition can be dominant or recessive. A dominant hearing loss or a dominant genetic condition of any sort means that it just takes a gene mutation from one parent for the condition to be realized. In a recessive condition, a recessive hearing loss, the same mutation needs to be handed down from both parents. Now, each parent can have a gene mutation that causes hearing loss, but unless it's the same gene mutation, the child will not have hearing loss. So it has to be the same gene mutation. And there are many of them that we know about, and still we believe many that we do not yet know about. There are also X-linked conditions, and this is a problem in the X chromosome, so handed down from usually from the mother. This condition tends to be, usually these conditions are, the phenotype is more affected for boys than for girls. So you've probably heard of Fragile X syndrome. And the reason for that is that boys get only one X chromosome, whereas girls get two. So girls have a better likelihood that if one of those chromosomes is not affected, that they will have a milder form of whatever the condition is. And then there can be mitochondrial disruptions, and the mitochondria that's a special part of the cell, and it's in the egg cells and not the sperm cells, and there are mitochondrial disorders. Okay, so let's look at the prevalence of genetic hearing loss. First of all, here you can see that this is telling us that about 1 in 500 children are born with congenital hearing loss. I think that's a little low. It's probably more like 1 in every 330, so 3 in 1,000. And then in terms of etiologies across these congenital hearing losses, 25% are idiopathic, meaning we don't know what it is. It can be anything. 25% are recognized etiologies, but are not genetic. So these would be things like viral infections, RH incompatibility with the mother, and so forth. Okay, so let's look. 50% of congenital hearing loss is caused by a genetic deficit. And of those, 30% are syndromic, meaning that there are other disabilities. So this would be something like Usher's syndrome, so deaf blindness, Treacher-Collins syndrome. And then 70% is non-syndromic. Of that 70%, as you can see, a large proportion is autosomal recessive. Okay, so again, both parents need to have the same recessive gene mutation for the child to have a hearing loss. We'll come back to that in a second here. About 20% is autosomal dominant. So this is what you see in families where really everybody's deaf. You might have seen Niall DeMarco on Dancing with the Stars, an actor who is deaf, and his whole family is deaf for many generations, and that's surely an autosomal dominant genetic condition. This across all children who have hearing loss, this is a very small proportion. 96% of children who are born with hearing loss are born to parents with normal hearing. And then a very small amount of genetic hearing loss is X-linked, and this is non-syndromic, okay? Mitochondrial hearing loss tends to fall into the syndromic category. Okay, so let's come back to this recessive category of hearing loss. As you can see, half of this is what's known as DFNB1, and that is these connexin deficits. And you will be hearing, if you work with children with hearing loss, many of them you will hear that they have connexin deficits. You now know and understand that what that means is that there's a problem with the protein that creates those gap junctions that allows potassium to flow and reach the stria vascularis to repopulate the scala media with potassium. Okay, and this is a very, very common cause of congenital hearing loss. Okay, now let's look at these kinds of congenital hearing loss and what they mean for the individual. So non-syndromic hearing loss, again, an individual with a non-syndromic hearing loss is otherwise healthy with no other medical or developmental concerns. It's mostly sensory neural, and the majority is autosomal recessive, as we discussed. And approximately 50% of individuals with non-syndromic hearing loss have negative histories of hearing loss, and they have these connexin deficits, also known as GJB2. Okay, so the gap junction deficit. And of course, there are other genes that can also be responsible for hearing loss, for autosomal recessive congenital non-syndromic hearing loss. Okay. Non-syndromic hearing loss can also have a dominant genetic cause, and there are about 30 genes associated with dominant hearing loss. So there's no single source of hearing loss for individuals who come from these families where hearing loss is so prevalent. Sometimes specific genes result in specific kinds of audiograms, but that's not always the case. And again, inheritance can also be X-linked or mitochondrial based. And we still don't understand all the sources of genetic hearing loss. Okay, what are some of the other causes of childhood hearing losses? Again, viruses. There are many viruses that can affect hearing, and they can do so in 3 ways, okay? So first of all, a virus can directly damage the cochlea or the auditory nerve. A virus can instead just establish an inflammatory response in the cochlea, and that inflammatory condition damages the hair cells, for example. Also, a virus can increase the likelihood that there might be a bacterial infection, such as meningitis, that can get into the cochlea and damage hearing. Some of the viruses we worry about are cytomegalovirus, rubella, measles, mumps, herpes simplex, and HIV. Rubella used to be a very common cause of hearing loss in children. In the late 1950s and 1960s, there was a rubella outbreak. So many pregnant women came down with rubella, and as a result, their children had hearing loss. So just as a cautionary tale, parents now who are anti-vaxxers and fail to give their daughters the rubella vaccine, it might not hurt their daughters, but they might be putting their grandchildren at risk, because were these daughters to grow up and contract rubella while they were pregnant, they could then be, that could lead to hearing loss in the children that they then have. Okay, other causes of childhood hearing loss, prematurity and related factors. Many premature babies are given ototoxic drugs to prevent infection. As the term suggests, oto meaning ear, toxic meaning poisonous. So ototoxic drugs are life-saving drugs that can cause hearing loss. Again, it tends to be in those high frequency regions. Chronic middle ear disease. This is more common in developing countries where otitis media is not treated as aggressively as we treat it. Maternal use of alcohol or drugs, and now I will tell you, I always trip over this word, hypobilirubinemia. Okay, so this is when the infant has an Rh incompatibility with the mother. Okay. And keep in mind, as we saw in the article that we reviewed, problems can exist in auditory functioning that are independent of these raised auditory thresholds. And there can be other causes that I have not cited here. Okay, so that is all congenital hearing loss, the sources of congenital hearing loss, possible sources, and we'll be talking about how we treat that in a minute. But now let's talk about age-related hearing loss. Getting old is a very common source of hearing loss. Almost everyone who is fortunate enough to age encounters a hearing loss. High frequencies, here are mean audiograms that this investigator computed across many subjects based on the decade of age. So you can see here it is for women, and you can see that thresholds decrease, especially in the higher frequencies, as women go from being 50 to over 85. And you can see that hearing loss declines more rapidly and to a greater extent for men than for women. This is always the case. Why that is, we don't know. It could be a protective factor of estrogen. And this just shows the prevalence of hearing loss. So you can see starting around 50 years of age, there's a sharp increase in the incidence of hearing loss. This means that in your work, whenever you're working with anyone, certainly over the age of 60, you should just assume that hearing loss is present and work with the individual as if they have a hearing loss. Now, what are the sources of age-related hearing loss? Well, they're numerous. First of all, the inner hair cells themselves decline, the number of inner hair cells decline with increasing age. Okay, you can see these are the blue circles here. Here are the inner hair cells, and you can see that the number of inner hair cells declines. The number of cell bodies for the auditory nerve fibers in the spiral ganglion, they also decline. There are these little black circles here, the spiral ganglion cells. Okay, but you can see that what declines most, to the greatest extent, are the auditory nerve fibers themselves. Again, they become detached from the inner hair cells. Deafferation is the term. And so now basically even functioning hair cells may not send signals to the central auditory system with as much efficiency. Remember, every inner hair cell in a young normal cochlea has multiple auditory nerve fibers terminating on it. Well, if half those nerve fibers are atrophy, then the signal being transmitted is not going to be as clear. You will not have loudness scaling that's as good. Remember, loudness scaling depends on two things. An increase in firing rate of individual auditory nerve fibers, and then the fact that more auditory nerve fibers are recruited as the signal becomes higher in amplitude. If you don't have as many auditory nerve fibers to be recruited, your loudness scaling is not going to be as accurate. Okay. Now down here, what we see, this is a normal young cochlea. You can see the 1 row of inner hair cells, the 3 rows of outer hair cells. This is the cochlea of an older animal. And you can see how there's hair cell damage. And this was from, my understanding is quiet raised animals. This is the spiral ganglion in a young animal and the spiral ganglion in an older animal. You can see the decrease in the number of cells here. Okay. The endocochlear potential. This also diminishes, okay? And you will recall that the endocochlear potential is the difference in polarity between the endolymph in the scala media and the perilymph in the scala tympani, right? And normally the endocochlear potential is about 80 microvolts. And this is what is needed when potassium enters the inner hair cell. It causes depolarization of that cell, which is what sets off the firing of the auditory nerve fiber, right? Without such a high endocochlear potential, this whole system is less efficient. It does not work as well. It takes a greater stimulus to set off the firing of the hair cell, okay? So again, that's the endocochlear potential. It declines with age. And here's a little bit more information on it. And over here, this graph is quite interesting. Again, this is from the same study. These were quiet-aged animals. And you can see that as the endocochlear potential, that's what's shown here, as it declines, you can see that the amount of hearing loss in the high frequencies becomes greater. Okay. Presbycusis is the term we use for age-related hearing loss, presbycusis, and this decline in the endocochlear potential is one reason why there is presbycusis. This is referred to as metabolic presbycusis. Okay. Physiologic changes. So here you can see, you'll recall the green line. This is the normal tuning curve. And you'll recall that every auditory nerve fiber has very sharp tuning. They respond at low levels to a very specific frequency. But without that endocochlear potential, what you find is, and without the outer hair cells, that the tuning of these afferent auditory neurons, auditory fibers, these afferent fibers, the tuning curve becomes broader and it requires greater stimulation in order to fire. So loss of outer hair cells and that loss of the endocochlear potential, they have the same effects. There's a loss in sensitivity and a loss in spectral specificity. Okay, and so with that, of course, these two factors can negatively influence speech recognition. Okay, so now let's talk about, come back to children who are born deaf and remind ourselves of why it's so hard for a deaf child to learn language. And here I'm talking about spoken language, of course. And this is true even if deaf child has a cochlear implant, and perhaps especially if they have implants. Okay, so here we have a waveform for a sentence, and again, a spectrogram. We've been over what the waveform and the spectrogram show us. In this case, I'm going to use this as a model of what a typical normal hearing person hears. And this is what it sounds like.
Dumb shoes will sing.
Dumb shoes will sing. In my work, we often use these sentences that have correct syntactic structure, but the vocabulary selection is set up so that they're semantically anomalous, so that the listener cannot readily use real world context to recognize the sentence. Okay, so now in the case of a hearing loss, as we saw in the last slide, the afferent nerve fibers, right, the tuning curves broaden, right? We have broader tuning curves, and when that happens now, we might model that as something like this, where the signal is not as distinct, it is not as clear. So now when I play this, you will hear this.
Dumb shoes will sing.
And you say, well, wait a minute, I hear that just fine with those broadened tuning curves. There's no problem there. Well, you will recall we've also talked about speech recognition in noise, and the fact that if you are unable, if you don't have good sensitivity to temporal modulation and spectral modulation, then you cannot be listening in the dips in the background signal, right? So, and this is even more true for younger listeners. Children, as you now know, temporal modulation detection and spectral modulation detection develop through childhood, certainly up to the age of 10. So here we have speech recognition for the first kind of sentence I played for you, the unprocessed one, the nice clear one, for adults and for 7-year-olds. And they were the kinds of sentences I just played, syntactically correct, but semantically anomalous. And you can see how 7-year-olds do not have as good speech recognition as adults. No surprise. And this is in noise, I should say. This is at 0 dB signal to noise level. What that means is that the RMS level, the root mean square level of the speech signal and the noise are the same, right? 0 dB. Okay, now when we put those processed signals, the ones simulating broadened tuning curves in noise, this is what we get. Listeners of both ages do more poorly, but the 7-year-olds do even more poorly than the adults. You need to keep this in mind, because when you're working with children, if they're in the schools and you go in and you try to make the case that a child with a hearing loss, even if it's a mild hearing loss, needs an FM system, needs something so that the teacher wears a microphone and the child has a system so that they get a nice clear signal from the teacher. Some school administrator is going to say, oh no, I've been in there and the signal's just fine. There's no problem with hearing the signal and the noise. Well, you're going to have to, you can pull this figure out if you want. This is what that adult administrator with normal hearing is, how they're performing, and this is how that 7-year-old with hearing loss is performing. Okay, so quite a difference. So what are the changes in cochlear anatomy and physiology if you're born with a hearing loss? Loss of hair cells, it can be outer hair cells and inner hair cells. Remember, loss of outer hair cells, even if there is no loss of inner hair cells, can lead to a mild or moderate hearing loss, certainly up to about 40 dB. And it leads to a loss of clarity of the signal. There can be a change in the stiffness of the basilar membrane, meaning, again, that frequency representation is not as clear. and you have to present the signal at higher levels. So again, that diminishes that frequency selectivity, cuts off those sharp tips of the tuning curves. There's a loss of ribbon synapses. Ribbon synapses, this is a term that applies to the fact that there's deafferation of the inner hair cells, meaning that those afferent nerve fibers detach from the inner hair cell. They're the ribbon synapses between the inner hair cell and the afferent nerve fibers. Okay, and again, we've seen this before. Here are nice normal inner and outer hair cells. This is what they look like when there is damage. So again, sensory neural hearing loss leads to broadened auditory filters and reduced dynamic range, right? So now the loudest, the highest amplitude sound that one can hear does not change with the hearing loss. What changes is the softest sound you can hear. So now, as you will recall, those of us with normal hearing can hear a range of sounds between 0 dB and 140 dB. If you lose some hearing, now you can't hear 0 dB. Maybe you only hear between 70 dB and 140 dB. So the scale is shrunk. So you do not get as sensitive or as robust loudness scaling when you have a hearing loss. So again, the three problems you have a loss of sensitivity, a loss of clarity, and again, because you lose that loudness scaling, you're not going to be able to listen in the dips of the background noise as well. Okay, now, of course, we can give a child a hearing aid, which improves sensitivity. But when the hearing loss is profound or severe, we might provide the child with a cochlear implant. So let's just talk about what a cochlear implant is. This is what they look like now. This is the external part. This is worn on the ear. This is the microphone here. And I want to emphasize this sits right in the ear canal because the goal is to provide a signal as close to what someone would be getting with normal hearing as possible. And as we know, the ear canal, that's the place where we hear. And if you have two cochlear implants now, you'll get the same interaural intensity and time differences. You might also get some of that, the filtering of the ear canal. And then this is the processor that sits externally. This is the part that is implanted under the skin on the scalp. And then this is what goes into the cochlear, wraps around the scala tympani in the cochlea. Okay? Okay. So again, this is what it looks like when it's implanted. Here's the external portion. Here's the headpiece. This is the internal portion. And so it is implanted. It's a very easy surgery. The electrode array comes down. It goes through the middle ear into the round window and through the scala tympani. It does not go all the way through the cochlea because the cochlea narrows too much at the end. It goes about 2/3 of the way through. Now, how does it work? Okay, well, the microphone sends the signal to that speech processor, and there's what's known as automatic gain control. So the level, very soft sounds are made, there's a lot of gain. Sounds that are not so soft, there's a little bit of gain, so that the signal now is within a truncated amplitude range. Then that signal is sent to a filter bank. So this is bandpass filter 1, low frequencies, maybe this is frequencies 300 to 443, and then this is slightly higher frequencies. Setting the range for each one of these channels is known as mapping the cochlear implant. So what the signal processor does then is to get out the amplitude of the signal in that frequency range, in each one of these frequency ranges, okay? So you get out an amplitude signal for this bandpass filtered range, and this one, and this one, and this one. And then that signal, each one of these signals, is sent down to the electrode array in the cochlea to the electrodes, and the electrodes are driven. The voltage is set according to the amplitude that was derived from the speech signal in that frequency range. Okay, and so then the electrode just provides an electrical signal in the region of the basilar membrane, and hopefully that signal reaches the auditory nerve. Okay, so that's how a cochlear implant functions. When children are given a cochlear implant, again, the surgery is actually quite simple. It can even be outpatient if they're in the hospital, for less than 24 hours. The child is kept quiet for a week, and then in 2 to 3 weeks, they have a post-op check, and then at 1 month, they come in to get the implant mapped. Now, let's look at what the signal is like through a cochlear implant. Again, here we have this nice, clear signal, what you would get if you have normal hearing. Here's this sentence.
Knees talk with mice.
Now, let's simulate what this would look like if we run it through a cochlear implant. Okay, you can see that there's great smearing of the speech signal. And this, as best as we can simulate it, this is about what it's like. Okay, so much less clear. So now if a child gets a cochlear implant, they have the problems associated with sensory neural hearing loss that we've already discussed, but you add the implant and it means that spectral resolution is further diminished. And one thing that I did not talk about is that, or that simulation does not replicate, does not simulate, is the frequency to place mismatch. Because the electrode array cannot go all the way to the end of the cochlea, it means that now where the signal, say the 200 hertz signal is being delivered, is a region of the basilar membrane that might typically code 700 hertz. Okay, so there's a mismatch between where the signal of a certain frequency is being delivered and the frequency that is typically coded by that point in the basilar membrane. So, a cochlear implant, it's remarkable that any listener does as well as they do with a cochlear implant. That's evidence that we hear with our brain. So what can we do about it? This is just a little simulation showing you that that often, as we know, listeners with even severe hearing losses have a little bit of hearing remaining in the low frequencies. They still have some hair cells that survived down at the apical end, the very tip of the cochlea. So what we can do is we can amplify that hearing with a hearing aid and use it as a supplement to the cochlear implant signal. What I'm going to do here is to play that implant simulation again, then play just the low frequency signal for you. That is the signal under, the acoustic signal under 250 hertz. Then I'll play them together. If you have sufficient low frequency signals on whatever transducer you're listening through, you should be able to hear that adding this frequency that is uninterpretable on its own aids intelligibility of the implant simulated signal. And then I'll play finally what the sentence actually was. Okay, so I'm going to go through these 4 steps for you. Here we go.
What time do you want to go to dinner?
Okay, so I hope that was a demonstration that adding that low frequency signal can really aid speech recognition through a cochlear implant. Typically cochlear implants, the lowest frequency that is coded on a cochlear implant is about 250 or 300 hertz. Okay, we can also add the visual signal. So now what I'm going to do in this demonstration is you'll get to see this fellow say a sentence with no visual signal. Then you will see it with the visual speech signal, and then you will see it with both the visual signal and that low frequency signal supporting the cochlear implant signal. Okay, here we go. So the point of all of this is that if you ever find yourself in a position of working with children with cochlear implants, you want to advocate for the child always being able to see the speaker and, when possible, have that low frequency signal added to what they're getting through a cochlear implant. Now, do cochlear implants really help children learn spoken language? Well, as a demonstration of that, what I'm going to do here is, first of all, play for you a sample of speech from a deaf child from the days before cochlear implants. These are some children I taught many years ago. These are their little hearing aids on their chest here. We were on our way to gym, so they're showing off their muscles there. And so you'll hear how they sounded, and then I'm going to play a sample of speech to you from a child who's the same age but got a cochlear implant at 1 year of age. In both cases, they're going to be talking about games they like to play. So see if you can understand what this child is saying first.
We have a game after today.
Okay. Do you like soccer? It is a fast sport. I am on a soccer team. We have a game after school today. I'll play that again for you.
And I'm on a soccer team. We have a game after today.
Okay, so these children were about 5, 6 years old. So now here's a little girl who's the same age, but got a cochlear implant at 1 year of age.
So how do you play Duck, Duck, Goose? I don't remember that one.
It's just regular Duck, 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 Goose.
Yeah, I'm not sure how to play, though.
You're not supposed to have just one or a few kids, like 3.
Mm-hmm.
But you want to, you can. You have to make a big circle, and one has to stand out, and you have to say duck, 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 goose. And then whenever you want to, you can say goose and duck, duck, duck. And then you say goose. That means the other kid that's down sitting.
Uh-huh.
They have to get up and try to tag them, and then they have to, and then if they tag them, that means the person that just did the duck, duck, goose, it has to go again, and then how you don't do it anymore, you have to sit in their spot. And then they do it, and then like another one, and then another one.
Oh my gosh.
Okay, so pretty good. Quite a difference, huh? Cochlear implants have done so much to help deaf children learn spoken language. But just to put this in perspective, here's a sample of a little girl with normal hearing giving the same sort of narrative. Tell us about a game you like to play.
Do you like take gym at school or PE?
PE.
PE. What's your favorite game you ever played in PE?
Scooters.
How do you play that?
See, there's like these little square things with
Okay.
a handle, and they're like, I think there's green, purple, yellow, and blue.
Okay.
I know there's purple, yellow, and blue.
Okay.
I'm not sure about green.
Okay.
Yeah, there's green.
Okay.
There are those colors, and every single one of them, no matter what color it is, it has a handle, and the handle is the same color as the thing.
Okay.
And they're called scooters.
Okay.
And like you need. This is how we always played with it.
Okay.
Each person has a partner and a scooter and a jump rope. And one person sits on the scooter like this,
Okay.
holds onto the rope, and the other person gets inside the rope and pulls. And pulls the other person on the scooter.
Oh my
And like
gosh.
all around, and it's really fun.
Okay, so perhaps you noticed some subtle differences in language. This little girl had more of what we call literate language and mental language. She said, I think, wait, there's yellow, green, and blue. I think there's blue, right? So she did that. And it was a little more elaborate, every single one of them. Okay, the first child with the cochlear implant had perfectly serviceable language, but it was not as flowery, it was not as enthusiastic, it was not as complex as this little girl's. So cochlear implants have done so much to help deaf children learn spoken language, which is important because spoken language is the basis of reading ability, right? Okay, but we still need to be supporting these children to help them reach their maximal language abilities. Okay, so let's draw this lecture to a close. Speech perception and hearing loss. What have we learned? The primary cause of congenital hearing loss is genetic. Age-related declines in hearing acuity are common, and we saw all the reasons for that. Hearing loss creates several problems in auditory function beyond a loss of sensitivity. Hearing aids can resolve the problem of sensitivity, but cannot improve the clarity or the problems of speech recognition in noise. And cochlear implants have allowed many patients with severe to profound hearing loss understand speech, but it does not provide a signal equivalent to the signals you get through natural hearing. Okay, so I will stop here if I can. Here we go. And I will thank you for listening and for being in class. And thank you and good day.